Well, hello and welcome back to APHA's 15 on COVID-19 series. I'm your host, Dan Zlot, and today we're going to be talking about accelerated COVID-19 vaccine development. We've been getting a lot of questions, as I'm sure many of you have, about how we were able to develop and deliver COVID-19 vaccines so quickly. So today we're going to take a look at that process and hopefully help provide you with some information that can help you answer questions that you're getting from your patients. So without further ado, let's go ahead and dive in. Our disclosure for today, this project was funded by a collaborative agreement with the Centers for Disease Control and Prevention, and the collaborative agreement number there is in the footer for you. And here are your learning objectives for today's episode. All right. Well, I think in order to understand the accelerated process, we first need to do a quick high-level overview of the standard drug development process. So let's do that really quickly. So I really like this graphic. This is a great way to sort of summarize the different phases of clinical trials and drug development. So we start off with preclinical, which is usually several years in length. Then we move into phase one, and you can see there's only a few people there, and the typical duration there is months, maybe years. Phase two, you see a few more people, and you move months to years. And then in phase three, you see a lot more people, years to decades. And then phase four, where you sort of have ongoing monitoring and then lots and lots and lots of people. So a great graphic just to kind of give us a high-level overview. So let's go ahead and dive in and start off with preclinical. So as we start to talk about preclinical trials, there's a lot of stuff that goes on here. So these are mainly laboratory and animal-based studies, and what we're trying to do here is to first understand the disease process. So a lot of times we don't even fully understand what's going on with a particular disease. So that's the first step, is to dig in either through animal or cell models and figure out what's going on. Next, we want to identify potential therapeutic options, and that can be done using in silico or computer-generated models if we have a particular target in mind, or maybe looking at cells and starting to identify what we think is going wrong, what pathways are involved, etc. So once we identify potential therapeutic options, we start testing those. And so when we look at testing therapeutic options, that's usually done first in cell models of a disease. So you can see whether or not on a cellular basis you're getting the responses you want from your particular drug target or vaccine if applicable. And then you develop an animal model for a disease process. And this sometimes is very straightforward because the animal model already exists. And other times you actually have to spend years developing an animal model that accurately reflects the disease process that we see in human beings. So once you have a reliable animal model, then you perform some testing to see if the therapeutic option does what you want it to do, whether that's a vaccine or a drug. And here we're looking for a few things. Number one, efficacy signals. Does the drug or vaccine actually work? Does it prevent the disease or treat the disease that we're looking to treat or prevent? What types of toxicities pop up in animals? That's often informative as you start moving over to humans, and certainly something you want to pay extra close attention to. What types of starting doses should we be thinking about? Are they really small? Are they really large? And especially when you couple that with toxicity dose thresholds, that helps to identify the therapeutic window and gives us our starting dose when we look at moving into a phase one clinical trial. So that's a quick summary of preclinical work, and this can take quite a while, years, sometimes even decades. So let's move on now and talk about phase one. So in phase one studies, these are usually pretty small trials, typically, dozens, maybe hundreds of patients in a really large phase one trial. These are typically going to be open label, non-randomized, non-controlled trials. And what we're looking for in a phase one is basically the right dose to use as we start moving into phase two, where we'll look at efficacy. So the primary outcomes in a phase one trial are going to be dose finding primarily. And then in addition, they're starting to look at what types of adverse events or toxicities you see in this population. A couple things to keep in mind is that because these are very, very small numbers of patients, you can't really get any sense of incidence of toxicities, and any incident data that you can get is really, the confidence intervals are pretty wide because of how small the numbers are. Some secondary outcomes that we're looking for are pharmacodynamics. So is there any type of a dose-response relationship? Does the drug behave in the human the way that it did in your cell and animal models? Is it doing what you want it to do? And then efficacy. So just to drive home the point, in a phase 1 trial, we're typically not looking to see whether or not the drug works. We're looking to see what the right dose is, and as an added bonus, sometimes you might get some efficacy data in a phase 1 trial. And phase 2 trials is really our first look at efficacy. So these are usually mid-sized, so typically hundreds. A large phase 2 you might see thousands. That's pretty unusual. And these are usually open label, non-randomized, non-controlled trials, although sometimes you'll start to see maybe some element of randomization or some element of a control, whether that's a placebo control or an active treatment control. And the primary outcomes here is going to be efficacy and safety. So we've already decided on the dose from phase 1, and now we're looking to see, does the drug actually work in human beings? And then we're also looking to see what types of safety signals do we see, what types of adverse events, and what incidence of adverse events are we seeing? Is it 2% of the population, 50% of the population? Are these low-level grade 1 or non-serious adverse events? Are they grade 4, very serious adverse events? So that's really what we're looking for in phase 2. Secondary outcomes might include subgroup analyses. So we might want to look at women versus men, people with heart disease, people who don't have heart disease, etc. And then whatever additional exploratory outcomes that might be appropriate for the drug, disease that's being studied. So once we've completed phase 2, then we move on to phase 3. And phase 3 trials are usually very, very large. These are typically in the thousands of people, and they are typically randomized, controlled, double-blind, usually double-blind. On occasion, you'll see an open-label trial. The control group here can either, again, be a placebo or an active control. It just depends on the disease state that you're studying and whether it's ethically appropriate to give a placebo. And the primary outcomes here are very similar to what you see in phase 2, but we're actually, again, we've got a nice control group usually. So efficacy versus the control group, we want to see, is it as good or better than the control? And then, of course, we're looking to reinforce the safety data since we have a lot more people being exposed to whatever intervention we're making. Secondary outcomes, again, are very similar, subgroup analyses and specific populations of interest, and then whatever additional exploratory outcomes might be appropriate for the disease and the drug being studied. So that's a high-level overview of the standard drug development process. So how did we accelerate this for the COVID-19 vaccine development? So we'll start off with preclinical. So this is a nice graphic from the Reagan-Udall Foundation that sort of highlights the normal process versus the accelerated process that was used in the pandemic. So as I mentioned, we'll start off in this preclinical phase. How are we able to get this done so quickly when it usually takes years? And the answer is that we relied on some technology. So we were able to, using some of the modern genetic sequencing technology that's come out, to rapidly sequence and distribute worldwide the COVID-19 viral genome so that scientists everywhere had access to this. So we could start to identify potential targets, important pieces of the viral genome, etc. So that was huge. Another thing is that we were able to utilize and adapt vaccine technologies that were already heavily researched for other diseases. So cancer is where a lot of these technologies actually originated, but then they were also heavily used in recent years looking at potential responses, vaccines for Ebola, for MERS, for SARS, etc. And of course, this is a SARS virus, so it's closely related to MERS as well as to the original SARS virus. And so because we already had a lot of those vaccine technologies in development, we were able to grab onto those, use that background research to understand how they work, their safety profiles, etc. And then because we were also interested in these diseases, we also had existing cell lines and animal models for SARS, which again is very closely related to SARS-CoV-2 or COVID-19. So that really allowed us to accelerate this preclinical process instead of having to spend years. Those years had already been spent. So we were drawing on that research and we were able to spend months. So that's one of the key things here is all the preclinical work was light years ahead of if we were starting from scratch. So next, we'll talk about the clinical trial phases, so phase 1, phase 2, phase 3. How do we get those things done quickly? So again, if you go back to the original, we see that these typically take, months for phase one, months to years for phase two, and years, if not decades, for phase three. And yet we were able to complete phase one, two, and three in less than a year. So how did we do that? And the answer is with some intentional design around the clinical trial process. So the phases of the trials were designed to overlap. So if you remember from the previous slide, typically phase one, phase two, phase three go sequentially with no overlap. And that's because clinical trials are expensive. So you want to complete your phase one before you move into your phase two. Likewise, and when you move into phase three, you want to make sure that there's an efficacy signal from your phase two. So drug manufacturers, drug developers typically don't overlap their trials unless they have a strong reason to, because of cost considerations. In this case, it was really important that cost was not so much of a factor. It was more important that we arrive at the therapeutic outcome, in this case, a vaccine. So the phases of the trials were intentionally designed to overlap. And what I mean by overlap is that trials were designed to be moved into the next phase while the earlier phases were still ongoing. So for example, if you look at some of the trials, the phase one trials were actually designed as what we call phase 1-2 studies. So basically there are some predefined criteria within the trial protocol that as soon as those criteria are met, allow that trial to move from phase one into phase two while the phase one trial is still ongoing. So we have a dose that we think is going to be safe, and then we're going to move that into phase two. Sometimes that means using multiple doses in phase two and studying those for efficacy. Sometimes it means we know definitely what the right dose is from the phase one, and we can move that into phase two and study just that dose. So that's a big factor. The other thing is that by doing this, you can allow the applicable patients from phase one to be included in the phase two study. So that limits or minimizes the number of patients you have to accrue as you move from phase one into phase two. So that's really important. And one thing that's important to point out is that the criteria to move from phase one to phase two or from phase two to phase three within these combination or overlapping trials are predefined in the research protocols. And so those are reviewed by both the Institutional Review Board as well as the FDA in order to allow those types of trial designs to happen. Now, one unusual thing about the COVID-19 trials is that typically in phase one and phase two trials, you do not see randomization and blinding. However, because of the need to push these things as quickly as possible, randomization, blinding, and even placebo controls were used even in the phase one and phase two trials. So again, this is highly unusual, but it was done because we knew we were going to want this information quickly. We wanted to know if this would work, and we wanted to make sure that we had the appropriate control groups to compare against when we started looking at efficacy outcomes. So all of that really helped us accelerate the clinical trials process. Additionally, when it comes to the clinical trials process, the graphic here is just sort of some of the standard things that need to be done to get a clinical trial launched and off the ground. And so it takes a lot of time, as you can imagine. There's a lot of regulatory paperwork, there's a lot of staff involved, there's a lot of training involved. And so the world, because this was such an important thing for us to address collectively, invested significant resources, and that's time, personnel, and money in getting these clinical trials put together, launched, and to start recruiting patients. So that includes site selection and initiation. So if you've been involved in clinical trials, you'll know what a big deal that is. There's a lot of work that goes into getting a site set up, a trial site set up, training all the personnel, etc. And then once you have your site ready to go, then you start recruiting patients. So there's a lot of work that goes into letting patients know that the trial is open, getting the word out there about that, screening, making sure that patients are eligible to participate, and then actually enrolling them in the trial. So there's a lot of work that goes into that. Lots of lab work, lots of history taking. You have to collect really complete medical histories. And then the enrollment, you have to get informed consent. You have to make sure that patients understand what they're participating in, and then make sure that all the procedures for the trial are followed to a T. And that can take a lot of time. So much focus was put on this that this was able to happen in probably the fastest in human history. We were able to put all these things together. And then, of course, ongoing data analysis. So typically, the way this works is you wait till you complete a phase 1 trial before you do your full data analysis, or you wait until you hit a pre-specified endpoint to do a safety analysis. So here, because this information was so important, data was being monitored and analyzed almost in real time. And so that also makes a huge difference in terms of our ability to understand and apply that data. Now, let's talk about some of the other things that were done to help accelerate the delivery of COVID-19 vaccine to the public. So let's talk about the manufacturing phase of the COVID-19 vaccine. And you can see here, typically, the manufacturing phase follows the clinical trials and the FDA review. Here in the pandemic, you'll see that manufacturing was taking place during the trials, and we'll talk about that. That's highly unusual. So large-scale manufacturing of the vaccine products began while these products were still in phase 2, 3 clinical trials. And the reason that's usually not done is because of expense. So it is outrageously expensive to set up a manufacturing process for a new medication. So before you invest in that process, typically you want to make sure that you have a drug that's actually been approved. And so here, because of the need to get vaccines delivered to people in an expedited fashion, manufacturers agreed to set up their manufacturing processes while the drugs were still being studied. So essentially, once we knew what the dose was going to be, they started the production of vaccines in large scale. And they were assisted by the US government in a number of different ways, either through pre-purchasing doses. So we saw that in the case of both Pfizer and Moderna vaccines, where the US government purchased hundreds of millions of doses before the vaccines were even fully approved. But that helped give Pfizer and Moderna some confidence that their dollars invested in the manufacturing process wouldn't necessarily go to waste. And then they also did some work around helping make sure that all the components needed for the vaccine were available. So that's how we were able to make sure that once the drugs were approved, they were able to start being shipped out relatively quickly after the FDA granted emergency use authorization. So now let's talk about the last phase here on this graph, which is the FDA review portion, as well as the continuous monitoring portion. So how do we make this happen more quickly than it usually happens? Well, the answer, very similar to clinical trials, is that the CDC and the FDA devoted significant resources, and again, that means time, personnel, and financial assets, to accelerating the receipt, categorization, analysis, and summarization of data. So if you have ever been involved in a new drug application, an investigational new drug, there are thousands, if not hundreds of thousands of pages of material to go through. So the FDA and the CDC groups that were responsible for reviewing all this information really invested a lot of personnel time to going through all of that information, summarizing it for the expert panels who were reviewing all this information, and then also in terms of prioritizing the regulatory oversight meetings. So as soon as the data was submitted, within 2 weeks, the CDC and the FDA were having meetings of their respective oversight bodies. So whether that's the ACIP group or the VRBPAC group, that helped make recommendations to CDC and FDA respectively. They prioritized ensuring that those meetings got scheduled in a very timely fashion so that the regulatory oversight process was not a holdup. And then, of course, once the vaccines received emergency use authorization, both the CDC and the FDA have been very proactive in active ongoing safety monitoring. So hopefully we've all heard a lot about the vSafe health checker program. So it's an app that can be downloaded on patients' phones for people to report any adverse events that they're experiencing after receiving their COVID-19 vaccinations. So that's how we're able to accelerate the regulatory oversight and the continuous monitoring phases of the COVID-19 vaccines. So what are the key takeaways? What kinds of messages can you give to your patients that hopefully help ensure that they can feel confident about receiving their COVID-19 vaccines? Well, the first key takeaway is that the COVID-19 vaccine development was faster because the scientific, medical, and regulatory communities made it their highest priority. Never in human history have we seen this type of coalescence of effort around a single priority like we saw with COVID-19 vaccine development. No safety steps were skipped in the COVID-19 vaccine development process. They're all there. The preclinical work was done, the phase 1, the phase 2, the phase 3 studies, those were all done. These drugs were studied in tens of thousands of people, and the ongoing safety monitoring continues. So no safety steps have been skipped. Only safe and effective COVID-19 vaccines that were rigorously tested in tens of thousands of volunteers received FDA authorization. And finally, COVID-19 vaccines are being held to the same safety standards as every other vaccine on the market, whether that's Shingrix, the flu vaccine, tetanus, it doesn't matter, all the same safety standards apply. So that will wrap up our episode of the 15 on COVID-19. We hope that this provided you some useful information to help you educate your patients and answer some of the questions that you're receiving, and we look forward to seeing you in our next episode.